Welcome to the Theoretical Neuroscience podcast. I am the host, David Dahmen. Before I got interested in neuroscience, I worked in the subfield of physics called solid-state physics. I made mathematical models to explore physical properties of semiconductors used, for example, to make electronic circuits. To model electric properties of the semiconductor silicon, one approach is to make a virtual silicon crystal. Here, virtual silicon atoms are positioned at the lattice positions of the silicon atoms in a real silicon crystal, and quantum mechanics, specifically the so-called Schrödinger equation, is solved numerically for the virtual crystal to calculate the physical properties of interest. These are difficult calculations to do because the Schrödinger equation for this system is complicated. But the major simplification is that all silicon atoms in the crystal are the same. It follows from atomic physics that all silicon atoms in the universe are identical. When we started making models for networks of neurons some decades ago, it was natural to assume the model neurons to all be identical as well. For one, it made the mathematical analysis easier. Also, one has to start somewhere, and the simplest starting point is to assume all neurons of the same type to be identical. That is, a so-called homogeneous population. After all, there are many ways the neurons can be different, but there is only one way the neurons can all be the same. These days, most network models assume several different neuronal populations, but neurons within a population are typically assumed to be identical. Of course, the scientists back in the days knew, like we do, that no two neurons in our brain are completely identical. Is this only because of the necessary noise and contingency in the development and maintenance of our brains? Or is some of the heterogeneity added by design? Does having a heterogeneous population of similar looking neurons have a benefit compared to having a homogeneous population where all neurons really are the same? Earlier this year, I came across the newly published paper, How Heterogeneity Shapes Dynamics and Computation in the Brain. Here, the 9 authors discuss the benefits and implications of within-type heterogeneity when it comes to dynamics, stability, computation, and self-organization. Today's guest is the first author of this paper, David Dahmen, a researcher at the Research Center Jülich in Germany. There is heterogeneity on essentially all levels of the brain, and we discuss this a bit before narrowing down on the heterogeneity of neurons. For very simplified neuron models, the problem of understanding heterogeneous neuronal populations has some similarities to a well-known system from statistical physics, so-called spin glasses. David has a physics background, and we talk about this link and how advanced techniques from statistical physics can be and are used in neuroscience. We further talk about how neuronal heterogeneity can be modeled, its effect on neuronal dynamics and neural computation, and also its clinical relevance. David has also worked on effects of synaptic heterogeneity, that is, effects of the variance of synaptic weights in a network. This is another type of heterogeneity, and we discuss how these two types of heterogeneity, neuronal and synaptic, interact to determine self-organization and learning. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking. For example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I really appreciate all of you who are already Patreon supporters. Thanks a lot. I will be most grateful if more join in to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community on social media or otherwise. Okay, here is David. So, welcome to the Theoretical Neuroscience podcast, David Dahmen.
Hi, Gaute. Thanks a lot for having me. It's a pleasure to be here.
Yeah, you are researcher now at the Forschungszentrum Jülich. That sounds really
Exactly.
cool. Forschungszentrum Jülich in Germany, or the research center in Germany.
Exactly.
And I guess you've been there for a while, and you've always been interested in problems at the maybe at the interface between neuroscience and statistical physics. Is that fair to say?
Yes, that's correct. So I'm a physicist by training. So I studied in Aachen and London, and then I moved to computational neuroscience for my PhD. It was under the supervision of Markus Diesmann in Jülich.
Mhm.
And then after the PhD, I switched the group to I'm now working with Moritz Helias more on theoretical tools and trying to basically import methods from theoretical physics to the field of neuroscience.
Mm. So this podcast is going to be about heterogeneity. Often you assume homogeneity, at least when you do modeling. All neurons are the same, or all something are the same. Not so strange, maybe, that we have this tradition, because if you sort of model a metal or something, you can assume that all copper atoms are the same. So it's not so. Yeah, it's not so strange that we actually maybe start with that. But anyway, so uh, if you sort of look at a computer, a computer is built up by transistors that are essentially all equal. That's at least the goal. So, and also, I guess the neural units in the networks underlying like artificial intelligence are also equal by design. So I think wouldn't it be a good idea if brains are built up by neurons that are equal? But that is not the case.
Yeah, that's a good point. So of course, for us theoretical physicists, it would be
Yeah.
much nicer if everything would be homogeneous.
Mm.
But yeah, so the question is this, let's say, heterogeneity that we see in neural networks in the brain, is this really something like biological noise or imprecision, or has this some deeper meaning that it basically can be used even for computation, right? So and I guess this is what we're gonna talk about, because there's
That's true.
evidence actually that it has a substantial impact on the network dynamics and also what these networks can then
Because
do.
I guess if you sort of were using electron microscopes to look into like the details of like a computer chip, you would also see slight differences in like metal contact points and stuff. And that would be if you started that they are not sort of relevant for, that's just noise in some sense, construction noise. So
Yes.
that's not relevant for the function. So I guess for the brain, it's not really clear whether it's a feature or like, yeah, whether it's the design thing or if it's just like accidental noise having to do with how the brain hardware works, sort of.
Yeah. I mean, it's definitely that it's clear that there are design choices, right? So, for example, what we like to do in neuroscience is to classify cells into different types, for example, right? And on the top level, there's of course the classification between excitatory cells and inhibitory cells. So those are
Mhm.
cells that basically depolarize all their postsynaptic targets, or hyperpolarize the postsynaptic targets. So there's quite a different function in these cells, and you can basically play this game even further, look at more and more properties like morphologies, other electrical properties, transcriptomics, so gene expressions, and so on, and you find more and more classes. But no matter how basically how fine-grained you define your classes, you still find a lot of heterogeneity even within the classes, right? And then this is a question: is this some residual noise, or also does this also have an impact,
Yeah,
basically?
yeah, yeah. I heard that. Well, I just heard that well, heard. I didn't hear. I read that Kepler speculated that snowflakes were alive, or the snow crystals were alive, because they were all different. So this thing of things being like, well, but that's I don't think that's not a big scientific question at the moment. You can make different things, inhomogeneous things, without life. But obviously, all atoms in the brain are the same, and I guess all all molecules with the same constituents, I mean, same built up by the same atoms, are also the same. But that's where the homogeneity stops, I guess. Like even inside neurons, like everything is different, right? Gene expression, for example.
Exactly. Everything is different. So the distribution of ion channels across the membranes. And then how this basically affects overall properties of your cell. If you look at the biophysics, then basically you find time constants which vary. You find excitability levels that vary, and so on. So it's very heterogeneous what you observe there.
Yeah. So that's at the cell level. Also, if you look at the circuit level, there's not a clear structure there either, right?
Exactly. So if you just look at basically which neurons could be connected, right, by just the overlap, studying the overlap of axonal arbors and dendritic arbors or so, then you find many more potential connections than there are, right? So we have sparsity also on the local level in terms of connectivity. And then if we go further, if we study then basically the strengths of a synapse or so, that also varies a lot, right? So there's many properties basically also on the
That's very
circuit level that are completely heterogeneous and scary to
Yeah.
to a theoretical physicist.
Yeah. So exactly. I mean, and people, of course, people are interested in sort of the effects of heterogeneity also in networks outside the brain, other types of networks, and then they talk about it's like nodes and edges. What's our nodes and edges in networks?
Yeah, exactly. As I said, the brain is really a multi-scale network of neurons. Yeah, and we can depict this mathematically as a graph. And this graph contains nodes, which are the neurons in our case, or if we choose a different level of description, it could be also a population of neurons or a brain area. But I'm typically working at the level of individual neurons and synapses. So for me, the nodes are really the individual neurons typically, and the edges are the connections between the neurons, so the synapses. Yeah, and then one can basically study nodal heterogeneity or edge heterogeneity if you want, right? These are the two types of heterogeneity that we could consider then mathematically.
Here we're going to because you had this paper that came out this year, how heterogeneity shapes dynamics and computations in the brain, and that focused, I guess, on nodal inhomogeneity.
Exactly,
So that's
exactly.
the brain.
Yeah.
Yeah. So this was a very nice perspective paper written by you and many co-authors. So one thing that you start with is sort of like this feature of neural synchrony, how heterogeneity affects the neural synchrony. And so what is neural synchrony?
Yeah, so synchrony means neurons are spiking, right? So they're communicating via spikes, which are short electric pulses. And synchronization then means how are the spikes of different neuron organized across time, basically. So synchronization would mean that different neurons fire their action potentials in a narrow time window together, basically. So you could you could study different regimes. So you find networks where you have more asynchronous type of activity, so where you don't see so much synchrony across neurons. But then there are mechanisms that try to synchronize these cells. This is also not. This is not unusual for an interacting many body systems, right? You find many other systems. In nature, where the individual constituents synchronize, and therefore it's also a highly studied field in physics. So typically, one then studies for example, oscillators that have a certain intrinsic frequency, and then due to the coupling, these oscillators might synchronize or desynchronize and form different macroscopic states of activity.
So before we go into that, it's not clear to me why, if somebody tells me that my neurons, the spikes in my neurons are more synchronized than used than they usually are, is that a good thing or a bad thing, right? I mean,
Yeah, it's a good question. So it depends on the task that you define. Basically, what your neural network should do. For example, it's quite obvious if all the neurons would fire at the same time, then why do you need so many neurons, right? So so they're all doing the same thing. So it's
Also,
so
you would get like this horrible epileptic seizures. So
exactly.
this
These are
that's
these
a bad thing. Yeah. Mm.
these are these pathological states, right? So full synchrony is not a good idea, basically. But also having everything pretty much uncorrelated is also not a good idea, depending what you want to do. And of course, due to the sheer fact that neurons are connected, they are going to be correlated in their dynamics, right? And there's different mechanisms that could enhance these correlations, and other mechanisms that could decorrelate neural activity.
Yeah. But I mean, so correlation, I mean, synchronization, that's more on the individual's spike times, maybe, and correlation is more on when they talk about population firing rates, maybe, or firing rates.
Yeah. So this is what like many people like it to put it this way to distinguish this on this level, but really it's a matter of time scale, right? So level of individual spikes means just you look at a very narrow time window where neurons are being correlated or synchronized, right?
Yeah. Because I think when you think about pathological states like this this shaking, like which is a symptom of Parkinson's disease, that seems to be some kind of a like a unfortunate synchronization, maybe, or at least that you get an oscillation.
Exactly. That's an
Unfortunate.
oscillation that is pathological, that is creating this tremor. Yeah,
Mm.
exactly.
Yeah. Okay, so now to the, I guess, like the, if you want to study, networks of oscillators, the model that most people have heard about, or to the extent that people have heard about these models at all, it's this Kuramoto oscillator model. So what's the structure of that model?
Yeah, exactly. So that's a phase oscillator model where basically each unit itself is already oscillating. Yeah. And then there's a coupling between neurons that depends on the phase difference between the neurons, right? And so
Mhm.
if you have a short phase difference, then the coupling is stronger, for example. And if you just wire these oscillators up together, then you find very interesting patterns. So you find states of desynchronization, and depending on the connectivity, you can also find, so on the coupling strengths between the neurons.
Mhm.
If this is sufficiently large, then you also find that these oscillators synchronize. And even though they might have different intrinsic frequencies, yeah, so even though there might be heterogeneity in these oscillators, they can synchronize.
Mhm. So okay, so in that, so how old is this model? This must have been 50 years or something. I don't know.
Yeah, it's very old. It's one of the standard models of let's say interacting many body systems, I would say, and it has been well studied. So there's also the so-called Kuwamoto order parameter, which is an important quantity which basically measures the level of synchronization on the macroscopic on the macroscopic scale, basically, and that's a determinant that basically people try to calculate and use in order to characterize the states of these
Hmm.
systems.
So you mentioned that, okay, so I mean, if it's like a homogeneous, meaning that all these nodes, all the Kuramoto neurons are identical, then depending on the coupling strength, you can get synchronization and stuff, right? That is some kind of like global synchronization of the whole, say, maybe a ring of these Kuramoto neurons. And then you said that can also happen if you have like these nodes are slightly different, meaning
Mm.
heterogeneous.
Hmm.
But does the heterogeneity help, or does it make it easier to synchronize, or is it more difficult to synchronize, or?
Yeah, so that's a good question. So naively, one would think the more heterogeneous your system is, the harder it is to synchronize, right? And this is also typically what has been found for a long time. However, the story doesn't seem to be that simple, because actually you find conditions where some level of heterogeneity actually helps in promoting synchrony, and that's a quite non-trivial effect, because it requires you to study different, let's say, different statistical orders of activity. For example, it could be that your overall level of activity is not high enough to support any synchronization, and then in this case, heterogeneity might actually help you to lift the overall level of activity, and that then enables synchronization. So there are conditions actually where a bit of heterogeneity, some intermediate levels of heterogeneity, can actually be helpful for synchronization.
I guess most of these, at least these studies, are essentially done numerically. But are there sort of like analytical solutions of the Kuramoto model, or I guess the homogeneous case then? Or
so there are methods, analytical methods, and theoretical physicists working on this. So in general, when it comes to these many body interacting systems, it's quite hard, of course, to do some theory, and the more heterogeneous the system gets, the more complicated it becomes. So a field in physics basically that has been devised for this is called statistical physics, where you basically try to find an effective description of your very high dimensional system. So if you have many oscillators or many particles of your system that are interacting or so, it's typically impossible to track basically all the variables, right? The activity of all the neurons, the activity of all the oscillators, or the position and velocity of all the particles in your gas or so. But then the statistical field theory tools help you basically to move one level up to go to a macroscopic description of these systems and basically study them in terms of effective macroscopic observables or order parameters, as we call them in statistical physics.
Okay. So now I'm going to come to talk about statistical field. Statistical field theory? I have this beautiful book here that's been written by you and Moritz Helias. So because statistical field theory is not, it's sort of like is it like a generalized mean field thing? What is statistical field theory?
Yeah. So mean field theory, if you have heard the term, is basically one.
I had several podcasts on that already. So.
Yes. So that's basically one approximation level of statistical field theory. Statistical field theory is basically a set of tools that allows you to study these many body interacting systems in a systematic manner, because what you typically find is that you cannot perform your analytic calculations to the full exact extent or so. So you need to do approximation levels. And for example, mean field theory is a theory that approximates the input to a neuron by a so-called mean field, right? There's different variants of this, depending on whether you look at the input or at the input fluctuations and so on. But the point is you approximate this input, and by this level of approximation, you then gain some analytical tractability of your system. And statistical field theory is basically the surrounding framework where you can derive mean field theories rigorously and also calculate corrections to these approximations in a systematic manner. In many body systems, the systematic manner typically depends on the network size. Yeah, so you make use of the fact that these systems are very large. So you typically cannot assume they're infinite, but they are large. And then basically you can arrange your calculations in basically orders of your network size, and study which effects are basically being suppressed the larger you make your network. Unfortunately, the networks in the brain are not too small. So this is why statistical field theory is also a tool that can be applied to neural networks.
Yeah. So this was, this is something which was really taken directly from this spin glass physics, right? It was like Parisi, or it was like someone who developed this.
Exactly.
Got a Nobel Prize in physics.
Exactly.
So this is sort of like a quite advanced tools, I would say, that you sort of are imported to. I think this is actually, I think often there's some, if you have people like you who sort of like come from this, like theoretical statistical physics, like really, and that can import these techniques into neuroscience. I guess this was like a paper from with this Sompolinsky and co-authors back in who started this in the
E-
90s. But I guess
e-
you
e-
and Moritz are developing it further or using it.
Exactly. So there was this famous paper in 1988 in Physical Review Letters by Andrea Crisanti, Hans-Jürgen Sommers, and Haim Sompolinsky, where they imported basically methods from the spin glasses to neural networks. And actually, that that step was actually not too big, because these spin glasses are also many body interacting systems, right? And they have what's called disorder in their couplings, so heterogeneity in their couplings. So the spins are the same, yeah. So it's not that complicated as in neural networks, but the couplings are already drawn from some random distribution. And for these kind of, so this is what makes it a spin glass, right? And for these kind of systems where you have this disorder in connectivity, you need these spin glass techniques. You basically try to understand what is the typical behavior of these systems, and if they're large enough, the typical behavior is also what you typically observe, what you observe in a single simulation, let's say, of the system. Um, yeah, exactly these methods are then imported into neuroscience. And the important distinction, I would say, is not only, I mean, spins can point upwards or downwards, or you can also make them soft, right? So you can give them some continuous
So
variables.
spins, I mean, we are both trained as physicists, so we learn about spins, but it's really like you can think about it like a magnetic compass needles that can either point upwards or downwards, or it depends also on the there's like different variations of that. But in the simplest case,
Exactly.
like
So it's a model of magnetism, right? These little magnetic moments, and if they align macroscopically, then you get some magnetization. And if they don't align, then you don't get any net magnetization. And people have tried to study basically under which conditions these systems become magnetic, basically on the macroscopic scale, in what kind of states you then observe. And the important fact is that these magnetic moments, they communicate with each other by magnetic forces, the Coulomb forces, which is a symmetric interaction, which means that basically two spins are basically reciprocally talking to each other, which is completely different in the brain, of course, right? Because we have a postsynaptic neuron and a presynaptic neuron, so we have a directed flow of information. And so these methods basically had to be adapted to study basically systems with some asymmetric connectivity profiles, which we have in the brain. So that's basically, I would say, the largest step that had to be made, going from these spin systems to neural networks.
okay, so that's yeah, we're going to, I mean, we're going to come back later in the podcast to your own work using this technique to study sort of like not node heterogeneity but edge heterogeneity, meaning synaptic inhomogeneity in synaptic connections. But anyway, so but this Kuramoto model, of course, then you have this. So then you have the basic units are neurons that are essentially firing regularly to begin with, and there's a
Exactly.
question whether they say sort of they synchronize. But of course, there are sort of like neurons that fire without input also in the brain, but at least in the cortex, they are not typically sort of like intrinsically oscillating. So how can you translate this to more like normal, whatever, cortical neural systems?
Yeah, exactly. So in cortex, you typically don't find these heavily synchronized states. You find asynchronous, irregular activity. So there's different types of neuron models which we can use for neurons. Ones that can be mapped directly onto this oscillator type of models that we discussed, like these Komodo or theta neurons. So quadratic integrated fire neuron, for example, would be one option, and there's a large community nowadays that uses these quadratic integrated fire
So
neurons in
why? I mean, why does that? I mean, quadratic has something to do with sort of like the integrating fire neurons, but it's like has a different sub-threshold dynamics.
Exactly. So there's no linear leak term as you would have, for example, in the leaky gradient file term, but there's a quadratic term. And then typically what you do is you define your threshold of your neurons to be at plus infinity, and then you close basically the axis, and then it basically goes back to minus infinity after spiking, and then it basically goes back. So this basically allows you to close the circle and map these kind of neurons onto phase oscillators. And then you can use this theories and these powerful tools that people have come up with networks of phase oscillators, which actually
Wow.
allow you to have some exact macroscopic formulation of your system. So these are called, I think, next generation neural mass models nowadays. So it started with Montbriot and Roxin, the paper on PRX. And then there's a large community now, Richard Gast, who's also author on this perspective papers, who is using this, and also
But I
Simona
guess
Olmi, Tom Giacchini.
but
So
it must be then like this this integrated fire neurons. It must be in the case where they get some kind of constant input or something.
exactly,
So that
exactly.
is exactly.
So
So
that they have
it's
basically driven over threshold all the time, right?
all the
And
time.
then it
Exactly.
and then it's basically about the interaction of these. But as you say,
Cool.
this is not the typical case. So many models of cortical dynamics also use then different types of neuron models which do not show this oscillatory behavior, right? And then the question is for these types of neuron models, how do you calculate basically how the fluctuations, how the correlations, how the synchrony arises in these systems? And that's a bit more complicated then, because you cannot nicely map it to these phase oscillator systems.
But this, so if it, well, if you think about sort of, I mean, the stability, I mean, that somehow the heterogeneity can promote stability of functioning complexes. I'm also thinking about when they talk about like ecosystems in the wild. I think it's sort of, if you have like a hit ecosystem with many animals eating each other, so it's like a quite complicated ecosystem, that they are more stable. I also learned that if you have this, maybe if you go walk around with these smartwatches, right, and they can take your measure your heartbeats, and somehow if you have a high variability in this inter heartbeat interval, that's a good thing supposedly. Not exactly sure, or yeah, or it's at least it's not. You would think that the more regular, the better, but that's not the thing. So it just seems like this somehow the heterogeneity and whatever disorder or sort of like non-regular behavior is some kind of has some stabilizing effect.
Exactly, it promotes robustness.
Yeah.
And this is also what people study in neural networks. So some of my colleagues, for example, from this perspective article, Axel Hood and Jeremy Lefebvre, they've published quite a few works already on how the stabilization actually works. So and there, what you typically find, for example, think of let's have a simple example. Think of let's say a neuron becomes active in a threshold-like manner. Let's say if the input is too small, then the neuron then the activation is zero. If the input passes a threshold, then it becomes one. Let's say you have a step-like, heaviside-like function or so, right? And now what happens if you now basically distribute these threshold values where this activation becomes one, right? Then effectively on a level of the population, not on the level of an individual neuron, but on the level of the population, what happens is that you smear out these very sharp step-like activation function and you turn it into a smooth function, right? And that basically makes your system more robust because it's not so sensitive again to these threshold-like behavior, but on a macroscopic population level, basically you smoothen your system, and that simplifies or stabilizes your dynamics.
Because it's I mean, if you I mean, I gave a course in computation neuroscience, and first an early model to study in these courses is this leaky integrating fire model. And if you look at sort of like the leaky integrating fire model, which has this sort of like sharp threshold, then it sort of has this sort of like quite sharp onset. But if you add noise, I mean current noise, it's not It's not like inhomogeneous, or it's no inhomogeneity in there. But that also has this linearizing effect. So I guess there's a link between inhomogeneity and adding noise, isn't it?
Yeah, exactly. At the population level, what you find is that actually that this heterogeneities and this noise basically have a similar effect. Yeah, they both basically make your activation smoother and stabilize your dynamics. However, it's there's a difference still, because one type, let's say noise, stochastic noise, temporal noise or so, is not reliable. So let's say if you do many trials of the same experiment, this noise will change, right? So this is something that basically smoothens and stabilizes your system, but you cannot basically compute with it because you don't know it, right? While heterogeneity could in principle be more stable, right? And that allows you that basically you can still make use of the individual neurons. They are more reliable, and still on the population level, your system is stable.
Hmm. So also this, I mean, when I looked at some of these stability studies of networks, there's this elegant way at looking at this eigenvalue spectra, isn't it? So
Yeah, exactly.
what is that all about?
Yeah, so these neural networks are nonlinear systems of coupled equations that where you try to find stable states, fixed points. And then the question whether when once you've found such a state, you ask the question: is this a stable state or is this an unstable state? Meaning, for example, if you would simulate the system and not exactly start in the state, would the system basically evolve into that state, or would it run away from that state? That is basically stability, right? And what you do then mathematically in order to study the stability is that you look at a little perturbation when you start basically your system start away from the fixed point with a little perturbation, and then you linearize your system around that state, and then you study basically what is the linear evolution of of your perturbation. And that basically gives you what is called a Jacobian in mathematical terms. So it gives you how is the coupling structure, the effective coupling structure of your system. And then this coupling structure can be nicely studied in terms of its eigenvalues and the eigenvalue spectrum, because basically you have a linearized system which you can decouple then. And these eigenvalues basically tell you in all of the directions in your phase space that are relevant, well, how is the perturbation evolving?
Mhm. Yeah. And if one of the eigenvalues are, say, larger than unity, then it will sort of run away from the minimum and it's unstable, right? So,
Exactly, larger than unity. If you have a leak term at -1 or so, right? Then you study basically how then this runs away. Yeah, and connected. So you can learn many things by these linearization studies actually. So you can look at time scales of how your fluctuations basically evolve, and this has also a very long history. So there was already a podcast on the balanced state, I guess, right? So
yeah,
um,
that's true.
so so in the balanced state, people showed that networks have this fast tracking property, so that basically the whole system can track changes in the external input much faster than an individual neuron can do. And that is basically a network effect that can be understood in terms of these eigenvalues. That basically this eigenvalue gives you an effective time scale of your system.
Yeah. Huh. Cool. And then you also mentioned something about that heterogeneity. It's a difficult word. Heterogeneity promotes a trivialization of energy landscapes. Okay, so what is energy landscapes, and what is trivialization of energy landscapes?
Yeah, so an energy landscape is basically a tool how to understand what how your states of your systems are organized. So this also comes from physics, for example, from spin glasses. There you can really write down an energy function. So each configuration of your system is linked to an energy. And then what happens typically over time is that your system reduces this energy, goes to some kind of ground state or minimal energy. It could be a local minimum, it could be a global minimum. Yeah. So and this this idea of energies could also be translated to a certain extent to neural networks. It's not so super trivial, because neural networks are these asymmetric systems, and but let's say we have a symmetric neural network or so, then we have this energy landscape, and then we can study what happens over time. If we initialize our system somewhere, then it will run down basically this energy landscape until it ends up at a local minimum. And how these local minima are organized, this basically depends strongly on the connectivity matrix, on the couplings of your system. So you can really design couplings such that you encode different energy minima. This is, for example, what happens in these associative memories, these Hopfield models or so.
Yeah. And then heterogeneity changes the energy landscape. It makes them less hilly, or what does it do?
Exactly. So trivialization basically means that the number of stable points, so energy minima, reduces. And you can basically view this if you have a very hilly landscape, and if you increase heterogeneity, then basically these minima basically get washed out until you basically end up with a big minimum, which is more stable then, of course, again, right? And but yeah, you get less of them, right? So it's basically washes out your energy landscape. And thereby it makes it again more stable, right? Because an external signal to your system could drive you from one minimum to the other minimum, right? Could give you the speed basically to go over the hills and the energy barriers. And if you have less of these minima, and then a perturbation to your system comes, then typically you stay, or you have the larger chances to stay where you are, and not drastically change the properties of your system by ending up in another state, let's say.
In your paper, you sort of make this. You first talk about the sort of like the effects of heterogeneity on neural dynamics, and that's essentially what we have discussed until now. And then you switch to neural computation. And this question: what is the difference between dynamics and computation? Sort of what is, yeah.
Yeah, that's a good question. Of course, any computation is supported by the dynamics, right? So the question is basically, computation is always basically linked to what you want to do with it, right? With the dynamics. So for example, as I just mentioned, so you can compute with these phase transitions, right? So you can make use of the fact that the system has different stable states, has multi-stability, has different minima in its energy landscape or so, in order to remember what kind of stimulus there was, right? So for example, you know that a certain stimulus always basically drives you in a certain direction in your activity space and lets you end up in a certain state. Then this would be, we could call this computation, let's say, right? But it's basically just using the dynamics that you have, of course.
And since we are able to do this computation, we think that maybe the mouse is able to take. It's in principle the mouse could do the same thing. I mean, yeah, because it's typical computation. It's so often on the perspective on the people, on us, right? People measuring the data.
Yeah, of course. So we believe that the mechanisms are similar across different species, right? So if we can compute with multi-stability, then potentially also the mouse and the monkey and so on use the same techniques, and also artificial neural networks, of course, can also use these. So that's another point, basically, where computation comes in, because you can train networks, of course, to perform a particular task. And there people have found that heterogeneity also is helpful if you want to train a system on a certain input-output relationship. Let's say you put in a stimulus and you want out a certain outcome, then you turn you change your parameters of the system. And if you start from an initial condition that is heterogeneous, that is also beneficial typically for these systems.
Yeah. So so one kind of computation, I guess, is sort of if you sort of send in, say, an oscillatory stimulus signal, maybe from like a sensory, some kind of sensory system into a neural network, maybe in the cortex. and then maybe like an oscillatory input, and then you want the neurons. You would say if the neurons are able to convey the information in this the oscillation, the frequency maybe in the spike trains, that's sort of I guess it's signal detection.
Exactly.
So heterogeneity can sort of also improve signal detection. Is that
exactly? So, and one can view this also, for example. So if we take your example of an oscillation or so, then this is a certain time scale that is relevant, right? And you can quite easily think that if your system has a larger repertoire of time scales, and if you have components in your system that promote certain time scales, then you can have these resonance effects and so on, right? And this allows, for example, there's a paper by Luca Mazzucato, one of the co-authors, where he showed that if you arrange your connectivity structure in such a way that you have clusters of different sizes, and these clusters act as a single unit, which has a certain time scale depending on the cluster size. So it's beneficial to have these heterogeneous cluster sizes then to promote different time scales of your system. And that can then be used, for example, these timescales can then also basically pick up the parts of your incoming signals, basically.
So then you can think of it as like each of these clusters are sort of resonating to like sort of like different whatever.
Yeah, exactly.
Different resonators picking up different frequencies. Mm-hmm. And you mentioned there's also stochastic resonance, right? How does that come into play here? That's a different kind of resonance.
Yeah, that's typically by noise, right? So this is typically an effect by noise that if you have an oscillatory signal, oscillatory system, and you send in a certain frequency or so, then at certain and at a particular frequency, basically the system reacts the strongest, right? So that's one mechanism basically which you find in oscillatory systems, that they are most sensitive to particular frequency.
And then noise helps you. So maybe if you don't have enough input to get over the threshold by yourself or just by the pure signal, then noise helps you to get over, and it's like more likely to happen when the input signal is at the highest, even though it's sub threshold. And so then okay, so again, it's then I guess noise and this thing quench disorder both could work maybe, or
well, yeah, exactly. It depends on which level you look at. At the population level, actually, both have a similar effect. Yeah.
Okay. Yeah, but at the single neuron level, I guess that's yeah. Yeah. Anyway, we don't have to think about that now. But of course, now when we, I mean, a very sort of like popular and powerful way, it seems, to look at experimental data from many spiking neurons is in terms of these neural manifolds. Can you sort of, so how does heterogeneity affect that picture?
Yeah, so that's a good way, good question. So these manifolds basically mean that your dynamics evolve in subspaces. So if you have n neurons or so, then you have n degrees of freedom, n axis of your coordinate system, let's say, right? And then you find that activity doesn't really span all of these axes completely, but it's located in certain parts of your manifold, right? Certain parts of your space. Which is then called a neural manifold. And of course, these the structures, as we talked about, these energy landscapes, for example, they basically give you fixed point structures in your system. So these minima of your energy landscape, they correspond to particular states. So particular parts of your space where then the activity would be located. And the fixed point is, of course, just the simplest motif that you can get. You can get also limit cycles, which correspond to oscillations. You could get different kinds of other attractors, which means that you encode certain structures in your network that basically have an attractive force on your dynamics, so that you do not really span all your possible state space, but are located in a lower dimension manifold. And this is exactly what many people nowadays find in neural recordings. We have these modern electrode technologies where we can record at the same time from many neurons, and we still find that the activity is typically localized in a subset of these degrees of freedom. So the question is how do these subspaces basically come about, right? And there's different mechanisms. There's these fixed point structures, limit circles and cycles and so on. But there's also many effects on the connectivity side that basically encode these certain directions in your state space that have more variance of activity than others.
Mm. So and so that's the neural manifolds. And how does the heterogeneity affect these manifolds to make them?
Yeah. So as we discussed, so there's this effect of trivialization, for example, and there's other effects that heterogeneity can have on changing the energy landscape. So this means if you change the energy landscape, then basically how the system gets attracted by different regions of your state space changes, right? So, for example, if you stabilize your system, then you can get rid completely of multistabilities, of bistabilities. So this means that basically the system will not jump between multiple stable fixed points, but basically stay rather located at one of them,
Okay.
right? So and thereby you change basically the structure of these manifolds that you would find.
Because one thing, I mean, like I think this manifold or this attractor thinking has sort of also been used in the context of decision making, that you sort of are sort of at and some kind of a hill between two valleys, and each valley corresponds to a particular decision, and then you don't really. So yeah, then and then you end up in one of them when you make a decision.
Exactly.
But I guess the heterogeneity then can actually force you to end up with a particular decision, or change your decision. And in that
Yeah.
context, yeah.
Yeah, and it doesn't have to be at that extreme that you basically get rid of this hill completely, but it basically determines also how high the hill is. So what is actually the capability of getting stuck right in one of them? So you could view it also in terms of decision making as a way basically for the system to become more flexible to different choices, right?
So
So
maybe because we have a problem, I guess, in the world today with people sort of being stuck in with certain views and maybe not willing to change their opinions even when facts change. So maybe we have to,
to make
yeah,
them more
maybe,
heterogeneous.
yeah, so, so that they are more, yeah, so that we are, of course, it applies to all humans, some more than others, but so yeah, and then you mentioned, so how do you actually tune? I mean, you mentioned this, that sort of like in the paper, that you can tune this neural inhomogeneity and take you from say like an asynchronous to a synchronous regime. That's really like a high level, on a like a high collective level. But how do you actually tune this neural inhomogeneity? You have to, well, I guess you have to go down at the cell level and then do something like add some ion channels, or take something away, or so it's a typically on longer time scales, or at least not millisecond time scales, maybe.
Yes, typically these are, for example, via neuromodulatory processes or so. These are longer time scales, exactly. And of course, yeah. So the way the question is, how can we study this, right? How can we basically, in a deterministic manner, change these level of heterogeneity? That's one question. But what one can also study is basically, for example, through diseases or so, can we find out whether also the level of heterogeneity is altered in the disease? And one of our colleagues has published on epilepsy research and found that the tissue seemed to be less heterogeneous than it usually is. So. Yeah, but yeah, as I said, one can study diseases basically in order to see what different levels of heterogeneity do to the function of a network. one can try to modify this. This is, of course, easiest in vitro. Let's say if you have a dish or so, you can look how it grows and study heterogeneity, or you can use some pharmacology to alter different effects. And there again, so let's say effects of neuromodulators can be very heterogeneous, because basically it depends on how the postsynaptic sites are basically responding to them, and depending on the receptors and so on, which are again heterogeneous. That these effects can be quite heterogeneous.
Oh. Huh. Interesting, because I mean, you could think of then in principle, if your colleague with the epilepsy findings, that it's like you need more heterogeneity, then you can imagine that is a cure or like a treatment involving then somehow making this more heterogeneous, right?
Yeah, that would be an option if this really turns out that it's not really. I mean, the alternative would be that, for example, a disease is targeting a particular type of cells, right? And that particular type of cells get damaged or so. But it could also be a general reduction in heterogeneity that is basically causing the problems. And then, of course, the question is how do you restore the level of heterogeneity? But if one would find that this is really the mechanism, this reduced level of heterogeneity, so that would be very interesting. So this is work by Jérémy Lefebvre and Taufik Valiante, so which I was.
But it really goes against our intuition, right? If you have some kind of machine problem and you come into the whatever machine room, and it's very messy, then you say, "Oh, this is not good. We should try to clean it, tidy it up." But here the intuition is actually it's the opposite.
This is the robustness that we discussed, right? The stability and in epilepsy, it's really this onset of these very strongly synchronized states that you would try to get rid of, right? And so it's making the system more stable, basically. What would help?
Okay, so until now we talked mainly about this node inhomogeneity, meaning that it's actually inhomogeneity in the neurons.
Mm-hmm.
And then you had this nice paper in a couple of years ago in PRX Life about edge inhomogeneity. I mean, brains actually, where it's like the synaptic connections, the connectivity, which can be more or less inhomogeneous, and how that affects the dynamics of the network. So what was that all about?
Yeah, exactly. So that's basically the part of heterogeneity of neural networks that has been studied longer, I would say, right? And this is why the perspective also focused on nodal heterogeneity, because it's basically the newer analyses. So this edge heterogeneity basically goes back to the spin glass studies in the 80s that we were talking about earlier, right? So this is basically a direct translation of what happens in these spin glasses, which have these disordered couplings, what happens in neural networks. And what we did in that study in PRX Live was basically deriving a machinery for understanding correlation structures in neural networks. And this basically came about by Sonja Grün in our lab in Jülich, having access to data or having data from macaque monkeys in motor cortex, recorded in Marseille by Alex Arile and Thomas Bauchier. And we basically looked at these correlations, and we also saw this large heterogeneity, this large diversity, right? So correlations, they are.
You saw diversity in the correlations.
Diversity in the correlation, exactly.
Compared just how similar the behavior of two neurons were, and you looked at different pairs, and it was very diverse.
Exactly. On average, neurons were pretty much uncorrelated. So across all the pairs of neurons, you find that the peak of the distribution is close to zero. And that has been long understood in terms of the balanced state that we talked about before, right? So that if you have some excess inhibition in your system, then this decorrelates your activity on average. But then nowadays we have all this information from the experiments, and we can have these very hundreds of neurons recorded in parallel, and we have this whole distributions. And the idea was basically what can we make of this data apart from understanding the mean that is very low, then that gives basically motivates that these networks are operating the balanced state. So the aim was really, can we learn something from this heterogeneity in connectivity and relate this into to heterogeneity in the coupling structure, in the effective coupling between neurons. And that was basically, so this PRX Live paper was basically the mathematical background of a paper that we published before on PNAS in 2019, where we did this data analysis and tried to really infer the heterogeneity level of the connectivity from the measured heterogeneity in covariances. And
so then you started with, to make this connection, you started with a model system.
Exactly. Yeah. So we made use of a study that was published earlier by Dimitro Gritsky and Moritz Helias and Tom Tetzlaff and others, that basically showed that that you can linearize certain nonlinear systems and understand correlation structure in these neural networks. And one of the studies you were also co-author, I believe, right? So.
Yeah.
So we know basically in certain asynchronous irregular states, we can linearize these nonlinear dynamics around these fixed points as we discussed before, and study basically these linearized perturbations to the state using these eigenvalue spectra of connectivity matrices. And yeah, that works for binary neurons, that works for stochastic point processes, that also works for leak integrated and fire neurons. And this is exactly what we made use of in this PRX Live paper. So we made a linearization of the whole dynamics around a stable working point, and then studied the fluctuations and the correlations in these fluctuations, and then compared this to spiking network simulations of these leak integrated and fire networks.
And then you actually found out that these like the variation of the covariances in the recordings, the spikes, is directly connected to the variance of the connectivity matrix.
Yeah, exactly. So
a strong finding, right?
Yeah. So maybe I have to explain it in historically how it came about. So these earlier works basically give you a relation between the covariance structure of your system and the effective connectivity structure of your system. So it's basically a big matrix equation that tells you how are the covariances organized if you have certain couplings, right? And that's of course a very powerful tool and powerful equation. It holds in these asynchronous irregular states, as I said, so not in all states, but for certain recordings of activity, one could assume that these assumptions are being met. But then the problem is, if you have this very powerful machinery of directly relating covariances to connectivity, then you also end up with a problem, because you can never, or with nowadays techniques, let's say, you cannot record from all the neurons, so you do not have all the covariances in your system, and neither do you have all the connections. So basically, although this machinery is very powerful, basically it's pretty much useless, because it's relating all the covariances and all the connections, and you don't know all of them, right? So even if you want to, like, for the inverse problem of inferring connectivity from measured covariances, you cannot really use it. So then the idea was, okay, maybe we need to be less ambitious and not really try to find out each and every link and each and every connection basically in these systems but let's say we only want to find out the statistics of the connectivity, because we had these distributions of covariances, and you can imagine if you would record now 10 times more neurons or 100 times more neurons or so, and if you don't have any strong bias in the subsampling, then this distribution would rather stay the same, right? So the mean and the width of this distribution or so should be quantities that are also representative for the larger circuit that we have not recorded. Right. So the idea of all this machinery was basically to move from this level of individual connections and individual synapses, individual covariances, to the level of statistics. So we have measured statistics of correlations, mean and variance of this distribution. What can we learn about mean and variance of the connectivity, so of heterogeneity level, basically? And so that yielded quite interesting results, because as we said before, so if you look at stability of these nonlinear systems, then basically these eigenvalues and their magnitude basically matters. So if you exceed a certain threshold, then your system becomes unstable. And what we found in our analysis is actually that these systems that we measured in motor cortex, and also in later studies we also measured them in mouse visual cortex, they seem to reside close to this point where actually the instability would occur. And that gives them interesting properties.
Because you were sort of able to conclude that that I mean it's that the connectivity, the variance in connectivity alone is able to explain your observations. You don't really need any variability in neurons, or
Exactly.
Yeah.
In that case, we didn't really look at variability in neurons. We said, well, variability in connections is sufficient to explain this. And then there were later studies, for example, by the group of Brent Aron. So we also looked at dimensionality of manifolds, how they are shaped basically by the heterogeneity in connectivity, because manifolds are basically determined by the correlation. So there's a link. And then, for example, the group by Brent Aron showed that there's more to it. It's not just connectivity, but also the firing rate levels of the neurons. So some kind of neuronal heterogeneity also has an impact on dimensionality and correlation. So definitely this is not basically the end of the story, what the relation that we found. But I guess it's still an important relation, because we see that this heterogeneity is very strong in edges in synapses, right? So alone due to the sparseness of the connectivity and then also synaptic amplitudes that are distributed in a very broad fashion. So just having this level of heterogeneity gives the system the possibility to have these very complicated and complex correlation structures.
I guess it's also, I mean, now it's one of the things that the field has learned to do is to record spikes from many neurons at the same time, with all these neuropixel probes and many neuropixel probes and so on. So actually being able to infer connectivity structure from these recordings is quite to infer heterogeneity from these recordings directly. That's quite important, right? Like
Yes, yeah, that's quite important. And there's many tools that are being devised that work on one or the other feature of activity. And for example, there's many tools that work on the cross-correlogram. So you look at correlation functions across time of neurons, and then you try to infer where is the peak. So is there a peak basically at positive time next or negative time next? And that gives you basically a hint whether there's a certain connection from one neuron to the other neuron, and which is the directionality of the neurons. So these are tools that are being devised. Personally, I'm a bit skeptical that these tools really work in all of the cases, because what we actually found in one of our studies in eLife was that in the state that I just described, where you have these very broad correlation structure across neurons, that these correlation structures do not only basically arise from having direct connections between neurons, because you can imagine that there's also effects that could basically hop across different stages of your network, right? So if you have very strong synapses, even though maybe it's not really on the individual spike level, but on covariances on longer time scales or so, then if one neuron fires, there's also a chance or an increase in firing probability of all the neurons that are not directly connected to this neuron, but also via indirect passes through the network. And this question came about by when we studied again this motor cortex data by Sonja Grün, which was recorded by a Utah array, which is spanning 4 by 4 millimeters of cortical surface and has 100 electrodes or so. And what we found there is, if we looked at spatial clustering of these correlation structures, is that you do not find strong correlations only in the close vicinity of a neuron, where you would assume that most of the local axon arborizations or so basically end. But you find basically a very large and salt and pepper-like covariance pattern all across the Utah array. And this we could then explain again with this heterogeneity in network connectivity that basically drives the system into a state where these indirect connections basically matter much more. And of course, if you have indirect connectivity, then it becomes much harder to infer from a cross-correlation function, basically the direct connections between neurons. Yeah. But in certain network regimes, that definitely works. Yeah.
But I guess one good thing is now is that there are more and more models now, like comprehensive, say, large network models that come that it becomes available. So you can, and even of course they are often they are sort of made to try to mimic a particular biological system. But even if they are not completely successful at that, they should be very nice, sort of like benchmarking. I mean, models to make benchmarking data sets to see if these sort of like analysis methods actually deliver as hoped.
Exactly. So that's a good test bed for all these methods. And
I think this is sort of a little bit surprised that our field is not using this more, because it's I mean, with all these new analysis methods, to have like sort of like fairly biologically plausible test data generated by these models is sort of like should be like a first sort of sanity test before they are applied on experimental data and well, drawing strong conclusions from experimental data. So that's maybe that could be done more, I think.
Yeah, you're fully right. Yeah.
Okay, so now we have talked about neural heterogeneity, and that was the first part, and now synaptic heterogeneity. And of course, this in the real brain, these things interact and occur at the same time, and not only that, but of course there's all of these like this homeostatic processes that determines like the ion channels in the neurons, and of course there's all this synaptic plasticity at different time scales. So how does all this work together?
Yeah, that's a good question. So it's a good point. So definitely, so these, for example, the synaptic plasticity, their activity, this plasticity is activity dependent, right? It depends on the, for example, in the simplest case of the spikes of the pre and postsynaptic neuron, for example, in STDP, short spike timing dependent plasticity. And of course, if you change the properties of one of the neurons by, for example, if you change electrical properties or so of these cells and makes them heterogeneous, that will have an effect on how the plasticity will basically change your synapses in the long run, right? So one would assume that these types of heterogeneity in neurons and synapses, that they are not independent, right? That they are correlated, and this is also what people nowadays start finding. Of course, this is very complicated because it's basically studying everything at once, or requires studying everything at once. But yeah, definitely this goes together.
Yeah. So there's lots of more things to figure out. But so how can you, like, the experimental strategies of studying neural heterogeneity? Of course, one thing is to actually look at individual reconstructed neurons, or look at like electron microscopy, and you can sort of look at the anatomy certainly, and also physiology, but to sort of look at the functional consequences of varying like the heterogeneity, and that is sort of harder, of course. But I guess so you mentioned is that you can do things in vitro, meaning like in cultures or in slices. So
Exactly.
what has been learned there?
So yeah, in cultures you have much more control, right? So you can in a very targeted manner you can manipulate cells a bit better. And for example, there what has been shown is that cells that are firing much further also have stronger connections to other neurons, right? So that the firing rate level also correlates to the level of the synaptic connectivity that these cells have. Yeah. But I think we are at very early stages in studying these things now. So cultures are one option, and looking at diseases is another option, to study how heterogeneity affects, and how basically on the level of individual neurons and synapses, how it correlates. There's still a long way to go, and one way, of course, where we have full control on how these things evolve is looking at artificial systems. So we can study network models, for example, as you said. But then, for example, certain types of plasticity, they happen on very long time scales. And if we then have very large networks, which we sometimes require to have because correlation structures are altered if we just downscale our system. And if we want to really study large models, as you say, which we nowadays have, and study plasticity in these models on longer time scales, then this becomes very tricky to simulate, right? Even on our largest supercomputers nowadays. So there is this field called neuromorphic computing, where people try to devise hardware that basically mimics the brain, that mimics features in the brain. So they really encode neurons in these systems as analog circuits. And there we have full control of what the model is, right? What the equations are, what we can measure the parameters, we can alter the parameters. And also these systems nowadays also support plasticity and learning, right? So we can use these systems as test beds, basically, on how heterogeneity evolves over time in a plastic scenario where we have where we can change all the parameters, and where they adapt also and change, emerge basically from the ongoing dynamics of the system. So this is a really nice test bed, as we argue in this perspective article, to study also the correlations between synapses.
Here then they are so much faster, right? So you can easily, and it's like orders of magnitude faster. So you can sort of simulate an hour where the on the real time it takes maybe to simulate a second on normal computers, right?
Exactly. It depends a lot on which neuromorphic system you look at. So there's different variants. And some of them really work with an acceleration factor of 1,000 or so compared to real time, because basically what you do is you encode really neurons in terms of their electrical components, like using capacitance, conductances, and whatever, and you make them tiny on these chips, right? So the values of all these parameters are so much smaller as they are in biology, and this gives rise basically to time scales, to time constants of your systems, which are much faster. In other types of neuromorphic hardware, the aim is not really to become so much faster than real time, let's say biological time, because for example, if you have robotics or so in mind as an application for these systems, then they need to interact in real time with the data that comes in, right? And there you don't need this acceleration factor of very large, right? So it depends, but definitely they can support these very fast simulations or emulations of neural networks. And that's, of course, a big plus, and also at a much lower power consumption than our supercomputers, of course.
Yeah, because I remember I was involved in like some of these large European projects, first brain scales and then the human brain project, where this neuromorphic like devices was one part. And I remember one of the, I guess a problem was that, or not a problem, but a feature of these neuromorphic devices were that, it was, you couldn't sort of control each parameter in, say, each neuron as accurately as you can do in a digital computer. So it was difficult to sort of like to make a direct sort of, direct sort of at this neuron by neuron comparison. But I guess when it's heterogeneous to begin with, it doesn't really matter so much, right? So they can be more useful for, and it's more the statistical properties that matters, right?
Exactly. So you make a big point. So of course, control is still limited on these devices of the. But yeah, if they come out of the fabrication process, they are typically heterogeneous, right? And then the question is, do we really need to try our best to fine tune the system afterwards, right? Or do we just embrace that heterogeneity and use it, right? And I think many strategies are nowadays basically trying to basically just accept this heterogeneity, say, well, the brain is a good role model that you can compute with heterogeneity, so let's just use it, and then basically try to find ways how to wire up your networks and how to use computation, for example, using these multi-stabilities or so, that are in line or that are even supported by these types of heterogeneities. Exactly.
I think in terms of network study, model studies, I mean, I guess the first generation of network models, they typically assumed all models, all neurons to be the same, or at least all neurons within a population to be the same. And it's of course that was a natural assumption. And of course, it's also about how to there's there's only one way to make all neurons equal. But if you want, there's like an infinite number of ways you can make a heterogeneous population. So in order for people to focus on something well defined and having a community trying to understand one well defined system, it's natural to start with a homogeneous system. But now I guess we have to sort of to be more. It's like a change of mindset. You think to go more into thinking about thinking
Yeah.
in terms of neurons in terms of like distributions rather than as some kind of mean representative.
Definitely, I think so, and you're making a big point. So these models that you were talking about, so they were also basically they were they were constrained often on terms of, let's say, population firing rates and like macroscopic observables, where some of the effects of heterogeneity might even not be so super visible. Some of them might average out, as we learned now. Some of them don't. Some of them change completely the structure and the attractor structural energy landscape, as we discussed, and so on. So it's very important to keep track of them, and also it is important to really validate these models on a larger number of observables, because what we found in these studies on this edge heterogeneity, for example, these distributions of correlations also, right? So you can devise many models that show this mean of covariances close to zero. These are all balanced models, right? But then there's still a huge space basically within that realm of balanced models, where you can tune the heterogeneity and so on. And then you need more measures to compare with, like this distribution of covariances. And then this tells you more about heterogeneity, or you can look at the dimensionality of these manifolds, and this constrains your networks even further. And then, yeah, it's really you should think of these properties of these networks in terms of distributions, and also not, as you said, not in terms necessary in terms of independent distributions, but these parameters and these heterogeneities can be correlated, right, across neurons and synapses, but also within synapses, right? In what we also find in experiments now, if you look at connectivity studies, people find more and more fine structure in connectivity that is beyond just population X is connected to population Y with a certain connection probability or so, right? So there's network motifs, for example, that you find, and all these kind of correlation structures, and basically that make up all these statistics that define these cell types and connection types. This should be taken more into account in our networks, because otherwise we will not. Yeah, it will be hard to really understand where all these features of this massively parallel data that we can nowadays record from, where they originate from.
Hmm. I mean, now if you look at the AI and the networks used in artificial intelligence, I guess they're typically all assumed to be or designed to be the same. And then you have like the synaptic connections, which are trained. So I guess you end up then with heterogeneous synaptic connections. And I'm not quite sure can you sort of speculate on what you could sort of gain by having more what you would call nodal inhomogeneity or
Mhm.
heterogeneity, meaning that having the neurons are different. I'm not quite sure how easy it would be to, of course, now a big thing about today's AI is that they're so extremely good at building really really small transistors and putting an enormous amount of GPU units on the whatever,
Yeah.
yeah, on the same chip, right? So maybe it's just changes, makes it much harder to produce these things so cheaply, but it would be
Yeah,
interesting
that I don't know, but I'm pretty sure it will have quite large effects on the behavior of these systems. And this is also what we already learned in terms of edge heterogeneity, right? It makes a large impact where you initialize your AI networks before training. So typically, like the simplest initialization that you could think of is that you draw all your weights and connections in your network from a Gaussian distribution, right, with zero mean and some variance. And then what people found out is that depending on the value of that variance on the heterogeneity of your initialization, training is much more successful or not, right? So there's actually large parallels to what we see in biological systems. So where we have typically recurrent dynamical systems, which can enter, for example, chaotic states where you have a large sensitive, where you are sensitive to perturbations. You find that the same regime where you enter into chaos in a recurrent network, discrete time recurrent network, and in a deep feed-forward network in AI, for example, this regime of heterogeneity for the initialization is also the most beneficial for AI systems, because you could think of like a stimulus in your biological system keep like staying in your system for a long time is the same as a data, like let's say an image that you present to an AI system, which can propagate through all these layers of these deep feed-forward networks, right? So there's a huge sensitivity of these systems on how you initialize them and what the initial level of heterogeneity is. And then I have to mention, so there's not only the weights that you train in this AI system, but each neuron also typically has a bias, right? And this bias you could view, if you want, as some kind of neural heterogeneity, because it basically shifts where your input of your system is with respect basically to your nonlinearity of your
it's a little bit like changing your threshold for
it's
firing.
it's a bit like threshold, yeah, that distributing your threshold. So also these things are being trained, but you're right. Like the typically the activation function that you use in these AI systems, that it's typically chosen the same for all the neurons in a layer or so, for example, right? And there's of course more options one would could have, right, in terms of distributing. And as we show as we saw. This matters really a lot on like also these distributed biases and so on. So, for example, Luca Mazzucato, who's also on this perspective article that we wrote, he showed that you can also train a network just by training the biases and not training the weights, right? So,
Ah,
like,
cool.
there's possibilities which you have and which basically enhance your capabilities.
Mm.
So I'm pretty sure that can be done more, and people are starting doing this, like integrating this heterogeneity in these AI networks.
Cool. So that's sort of that's my I have now covered my gone through my list of questions. I've learned a lot. So is there anything that you would like to that I have forgotten that you would like to?
No, I think I should just thank you. I mean, it was a very great discussion. I hope that I could give you a good overview of heterogeneity and why it's important.
Yeah.
And as you say, I think that this is a field that should be studied further in the future.
Yeah.
There's much more to explore.
Uh,
Yeah, and
it
I'll
just
look.
strikes me that we should have many more people in the field. There's so many avenues to doing this. So there's so many things that should be explored,
That's
and
true. Yeah,
so we are just like yeah, we need the influx of young, eager brains. That's true.
true.
Yeah. Very good. Okay. Thanks a lot, David. It was very nice to talk to you.
Thank you so much, Gautam.